Hello students. Today I am going to deal with the topic that is threats and risks of genetically modified crops. In this topic, mainly we will be studying about the advantages, not but the disadvantages of the genetically modified crops. Already you know what are genetically modified crops. In the golden rice topic, I said, or you all studied, that golden rice is considered as the genetically modified crop because, for the purpose of biofortification or just to increase the nutritive value of the rice plant, the genetic content or the DNA of the rice plant is modified. Such plants or such organisms whose genetic material is being modified in order to have the desired character in those organisms are said to be genetically modified crops. However, through recombinant DNA technology or biotechnology, scientists are developing new varieties of the plants or the animals or the organisms, but those all have, along with the advantages, some disadvantages also. So in this topic we will be studying about those threats or the risks of genetically modified crops as we are altering the genome or the genetic content of the plant. When you study this step by step, first aspect or the first risk of the genetically modified crop is that whenever we introduce a new variety or whenever we modify the genetic makeup of an organism, it leads to genetic contamination because of the process that is called as interbreeding. Sometimes what happens, crossbreeding also occurs between the two organisms, which will lead to what? Genetic contamination. So we will be discussing this point, that is genetic contamination or interbreeding or crossbreeding. So in this aspect, what happens, say for example, whenever we introduced a new variety into an ecosystem or in the field, that genetically modified organism may interbreed with the wild varieties or the endemic species. Endemic species means the plants or the organisms which are already native to that place are said to be endemic species. So here what happens, genetically modified organisms may interbreed with the wild varieties or the endemic species, and we call it as what? The crossbreeding, or you may call it as what? The interbreeding, which will lead to what? The genetic contamination. So genetic contamination means here what happens, the novel characters or the important characters that are present in the wild varieties or the endemic species may be lost due to crossbreeding. So sometimes what happens, whenever we introduce the genetically modified organism in an ecosystem, what happens, say for example, plant, a genetically modified plant. So we have introduced a gene. That gene is present in the pollen grain, and that pollen grain will go and it will fall on the stigma of the another plant. And one thing we should remember here, always the breeding occurs between the compatible species. So whenever that pollen grain goes and fall onto the other plant, what happens, the desired gene which we have introduced in the GM crop will go and get accumulated in the wild species and the endemic species, and that plant starts expressing the desired character, which we call it as what? The genetic contamination. Means along with the genetically modified organism, the endemic species or the wild varieties also gains the gene which we had inserted in the genetically modified crops. Because of this, what happens, the genetic material or the DNA of the wild varieties or the endemic species also occurs. Due to this, sometimes what happens, whenever a new gene is inserted in the wild varieties or the endemic species, the important characters or the novel characters that are present in the wild varieties may be lost due to the insertion of the new gene which has occurred to the crossbreeding. That is the first risk or the threat of the GM crops. And but in this slide you can find that, say for example, you have introduced an resistant gene to herbicides. You know very well, herbicides means these are the compounds or these are the chemicals which are used to kill the herbs. So you have raised a genetically modified plant which carries the herbicide resistant gene in order to kill the herbs. So whenever you have introduced the herbicide resistant gene, what happens, whenever you spray the herbicides to those plants which carries the herbicide resistant genes, they cannot be killed by the action of the herbicides. So now you have developed an herbicide resistant plant, and now you are introducing that herbicide resistant plant into the ecosystem or in the field. Sometimes what happens, because of the interbreeding or because of the cross pollination, that herbicide resistant gene may be transferred into another herb or another weed which is unwanted in the field. That time what happens, that weed also gains the herbicide resistant gene and that start growing luxuriantly in the field along with your crop. So whenever you want to remove that weed, you are going to spray the herbicides, but with the herbicides that weed cannot be killed, because through crossbreeding or through interbreeding that plant has gained the herbicide resistant gene from the genetically modified plant, because of which the herbicide will not have any effect on the weed. Because of this, sometimes what happens, the total field may be containing the weeds only, and the farmer is going to have only the less amount of the crop, or sometimes what happens, totally the crops in the field may be lost. These are all the points which we need to cover in the genetic contamination or interbreeding or the crossbreeding. And in order to kill the weeds, we need to add more and more amount of the herbicides or the weedicides. The herbicides or the weedicides usually which we use are chemicals. So whenever you start using more and more amount of the herbicides or the weedicides, what happens, this may cause the soil and water contamination. So these all are what the negative impact or the risks of the GM crops. The second risk or the disadvantage of the genetically modified plants or the crops is that horizontal transfer of genes to the other organisms, so which you are studying under the studying that HGT. So HGT means horizontal transfer of genes. So one of the example I would like to quote here, one of the genetically modified plant. So one best example which you know is golden rice is the genetically modified crop. Similarly, the another example for the genetically modified crop is GM tomato, called as flavor saver tomato. The character of this flavor saver tomato is that this plant will neutralize the effect of one of the enzyme called as polygalactourinose. We call it as what? PG gene, polygalactourinose gene. So what is the importance of this polygalactourinose? Polygalactourinose is an enzyme. This enzyme is produced by normal or wild varieties of the tomato, and the function of this polygalactourinase is that it is going to dissolve the pectin that is present in the cell wall of the tomato. Because of this, what happens, softening of the fruit occurs and the shelf life of the tomato is reduced. In order to overcome the softening of the tomato, or in order to enhance or increase the shelf life of the tomato, one of the tomato varieties developed, and that the tomato is called as flavor saver tomato. In this tomato, what is done, the antisense, by using antisense RNA, By using that technology, this variety is developed, and in this tomato, the gene is introduced in such a way that the gene is able to neutralize the effect of the polygalacturonase, so that this enzyme is not produced by the tomato plant. That is, polygalacturonase enzyme is not produced by flavor saver tomato. So you should remember, flavor saver tomato is unable to produce the enzyme that is polygalacturonase. So when this enzyme is not produced, there is no dissolution of the pectin that is present in the cell wall of the tomato, because of which the shelf life of the tomato is introduced. This is one advantage of the tomato. But in order to raise these varieties for the selection purpose in the laboratory, that is, selection purpose means for selection of the transformants, that is, whether the tomato has received this gene or not, in the vector or in the DNA or in the plasmid, the marker genes say antibiotic resistant gene is used. Means, you just remember, along with our desired gene, in recombinant DNA technology, the another gene is introduced that is called as the antibiotic resistant gene. The antibiotic resistant gene used in developing this plant is the kanamycin gene. So when this kanamycin gene is present, the plant will show resistance to kanamycin whenever we add antibiotic to the medium in the laboratory for the selection purpose. The tomato which survive in the kanamycin are said to be the transformants. That means they have received our desired gene. And the tomatoes which are unable to survive in the medium containing kanamycin are said to be non-transformants, because they are unable to overcome the effect of the kanamycin. So this criteria is used in developing the transformants. Whenever we use these plants in the field, sometimes what happens, the microorganisms, say for example viruses or the bacteria, have the ability to receive these genes. Especially in nature, we know that transformation is the very common phenomena. Transformation means, it is one of the replication of the reproduction which we see in case of bacteria, where transfer or the absorption of the naked DNA from the surrounding environment occurs in the bacteria. So sometimes what happens, the DNA of the tomato carrying the kanamycin resistant gene may get transferred into the bacteria, and that bacteria becomes, say for example, I said already, flavor saver tomato has which gene? That is the kanamycin resistant gene. When this kanamycin resistant gene gets transferred into the microorganism, say for example bacteria, if it is normal bacteria, not harmful bacteria, non-pathogenic bacteria, okay. Suppose if this resistant gene gets transferred into any pathogenic bacteria that is present in the environment, then what happens, that pathogen becomes antibiotic resistance, say for example kanamycin resistant bacteria. If that pathogenic bacteria which has already gained the antibiotic resistant gene, if it infects the human being, means it will show resistance to antibiotic, and thus it will have a very bad effect on the human health. So this way of transferring the genes from genetically modified crop into the microorganisms that are existing in the nature is said to be horizontal transfer of the genes, that is HGT. This is one of the most risky thing with respect to the genetically modified crops. In the next slide, the third risk or the threat of the genetically modified crop is that the genetically modified crops will compete with the endemic species or the natural species. Say for example, in the genetically modified crops, what we have done, we have introduced many genes. Say for example, if you have introduced the stress resistant gene, if we introduce that variety into the field, what happens, as it can overcome the stress that is present in the environment, it can compete with the the natural species that are present in the nature for the food or for the nutrients, and it will grow very invasive or luxuriantly. It will grow by absorbing the nutrients that are present in the soil, and it may inhibit the growth of the other necessary plants or the natural species that are present in the nature, because of which we may lose some of the important varieties that are present in the soil. Or I can say that is, later I'm going to explain you, loss of the biodiversity can be seen due to the luxuriant growth of the GMOs. That is, these GMOs have the ability of faster growth and can compete with the natural species and may cause ecological and the economic damage.